All right, let's take a look at the Human Genome Project. The Human Genome Project was a scientific process to map the entire human genome. And a genome is a full set of genetic information within an organism or virus. So basically, you take an organism and at nucleotide pair 1, is that an A or G, C, or T? At step part 2, you do that again, and then for humans, you had to do that 3 billion times. It took about 12 years to do this. So this is one technique, and this is another technique. I'm not really going to get into those techniques. Now, the findings is that we have about 21,000 genes. So we're humans on here. Right there. We have about 21,000 genes there, and we make about 80,000 proteins. All right? As I mentioned, we have 3 billion base pairs. That's what that is trying to show. So this is in billions of base pairs, right? All right. And so what we also find is that humans are 99.9% the same. All right. So I just want to point out, the difference between you and any other individual is 0.1, but there's still a lot of variation in that 0.1. So that comes about 3 million different possibilities. All right. Now, what we also found, though, is that most of our DNA is non-coding. Here's the exons. That's the coding region. Most of the other stuff is non-coding. All right? So introns, regulatory sequence, unique non-DNA, repetitive DNA. So repetitive DNA including, so this is stuff we don't know why it's there, but if a mutation occurs in a lot of this stuff, it doesn't affect you one way or the other. Now let's take a look at telomeres. Telomeres are repetitive DNA that's found in the end of a eukaryotic cell. All right? So what happens here is at each cell division, ends of the telomeres are going to break off. All right? So anytime we have a cell division, a little bit of that telomere breaks off. All right? So essentially what they are is they are buffers for coding regions of DNA. All right? And so what also this tells us is that cells can only divide a certain number of times before we start breaking off those coding regions of DNA, all right? And so when you break off coding regions of DNA, that leads to a mutation, and that leads to a cancer, which leads to death. And this is why clones have a shortened lifespan in comparison to individuals that are not cloned, all right? So Dolly lived to be only about 8 years old. Now, which is different from a typical sheep, domesticated sheep, which lives to be about 15. So by taking her DNA from an adult sheep, you took that DNA from a sheep that had shortened telomeres. All right? And so when you make a clone, you don't start off. So like if they clone me today, you wouldn't have an adult Brett Egger here today. What you would have is that you would have a zygote. And what would be unfortunate about that zygote is that zygote would have 49-year-old telomeres on it. And so that would greatly shorten that clone's life. All right, so let's look at, and so this is trying to show this here, the telomeres breaking off, and then, leads to cell death there. All right, here's a kid who he was born with this syndrome, which gave him shorter telomeres, unfortunately. So he ages much faster. All right, now let's look at the one gene, one polypeptide hypothesis. Now, before the Human Genome Project, biologists thought there were about 100,000 genes because we made about 100,000 proteins. So what that one gene, one polypeptide hypothesis stated was that for every gene, so for every protein that we had to have a gene for that particular protein. And what we found out after the Human Genome Project was that was incorrect. We have 21,000 genes that make about 80,000 proteins. We also found out why or how that is incorrect. And this shows us here is that we can have one gene that we can make different messenger RNAs with. All right, so here we have different exons, all right, and in some cases, some exons are cut out to make this messenger RNA, and a different exon is cut out to make this messenger RNA. So these proteins are going to be similar to each other, but not exactly the same, and this is how we can get more than one protein off of the same gene. All right. Let's take a look at the human immunodeficiency virus. This is a virus that attacks our white blood cells, our immune cells. Now, it's only about 9 genes long, all right? But, if you have my notes, the deaths on this annually have gone down because of better medications there. Now, let's talk about all diseases. We can take a look at all diseases and classify them 2 ways. One are genetic diseases, and this is a disease you have at fertilization, like cystic fibrosis, like sickle cell disease. And then there's also acquired diseases. And these are diseases that you get from the environment. So like polio, or measles, or mumps, or AIDS, or, COVID-19, all right? So we look at HIV to AIDS. So somebody having an HIV infection to having full-blown AIDS without modern medication. What would happen with this person is they would get full-blown AIDS in 6 to 7 years, or 10 to 12 years, or 2 to 3 years, or no infection. So the no infection group, all right, so before we get to that, we have to look at this. So HIV binds to 2 receptors. Remember, I told you a virus needs to bind to a specific receptor on our cells. So HIV binds to 2 receptors. These are known as CD4 and CCR5. All right? So that no infection group are genetically predisposed not to get an HIV infection because they don't make CCR5. And that's what we see here. And because there's no CCR5, HIV can't attach to their cells, and so there's no infection that can occur. So if we look at those other groups, the 6 to 7 year group, they have 2 genes for making CCR5. So they produce, several of these CCR5 molecules, all right? The individuals in the 10 to 12 year group, they only have 1 gene for CCR5, so they make half of the CCR5 that the 6 to 7 year group does. And so you can see, in order for a virus to infect a cell, the virus has to bump into that cell, all right? And when it bumps into the cell, if there's half of those CCR5s around, it takes it twice as long to infect that cell. And so that's why we see that length and difference here. Now, the 2 to 3 year group has a mistake in their promoter, and they make twice as much CCR5 as the 6 to 7 year group, all right? And so they get infected very easily then, all right? So when we look at this, and we look at how this came to be, is that in how we have these people who don't produce CCR5. Well, people of Northern and Western European descent, about 1 in 5 are heterozygous for not producing CCR5, all right? So that means about 1 in 20 don't make CCR5. So there was a bacterium that would grab onto CCR5 and then invade cells, all right? So they would get inside the cell and eat the cell from the inside out. And that bacterium is known as Yersinia pestis, and that's the bacterium that causes the Black Plague. The Black Plague killed 1/3 of Europe between the 1300s and 1600s, all right? So this bacterium used the same protein that the HIV virus does as well, all right? And so this is an example of natural selection occurring in humans. If you have the right genes for an environment, and this was not producing CCR5 in Europe during the 13 to 1600s, you didn't get this Yersinia pestis infection, and so you didn't die of the Black Plague. If you did produce CCR5, you died before you got to reproduce. And so this is why we see more and more individuals of Northern European descent having that gene for no CCR5. And that is the highest of any other ethnic group found on the globe.